Thank you so much for the opportunity to come and talk to you about some of our work. Of course, I just have to say that the work that I'm going to be talking about, I'm representing the team that works on this, but there's a large group of people that have all contributed and worked really hard to make this work happen over the last 7 or 8 years. I put my contact information up there. It'll be also at the end of the talk. Feel free to reach out and ask any questions. So just a brief outline. We'll talk about how liver abscesses are diagnosed and classified, look at some of the economic costs.
You can take the mic out of the stand if you want, if that's easier. Just pop it over. Hold it up to your face.
All right. Like a rock star now. So we'll talk about economic costs, both the seen costs that you see directly and unseen costs of liver abscesses. Some factors that influence liver abscess development rates, the theories on how they form and the microbiology involved, current approaches for preventing liver abscesses, and I'm going to touch on some of the challenges that come along with studying liver abscesses. So one of the issues is that you can't diagnose animals that have liver abscesses until they've been processed. Most of the time, cattle don't show clinical signs unless the liver has been very severely abscessed and is actually has its function impaired. To date, people have looked for they're called ante-mortem markers to try and identify which animals have abscesses and which ones don't. That hasn't yet been developed yet, but our data suggests that it might be possible to develop a blood-based test looking at gene expression. One of the other things that's been used is ultrasound of the liver to see if you can visualize any abnormalities in the liver, but it's quite hard to visualize the entire liver. So you might catch some of them, but some of them might be invisible to the ultrasound. So one question, I've got some feedback from producers about this, is there actually value in diagnosing an individual animal with a liver abscess prior to slaughter? So if you have thoughts on that, feel free to let me know after. So abscesses are classified based on the Elanco liver scoring system. Let's get our pointer. So this is a healthy liver. There's no abscesses, so this would be a 0. Livers that have 1 small or 2 small abscesses are classified as an A. And those that are severely abscessed, so they have multiple small abscesses, they might have a very large abscess. Some of these livers, the entire liver can be a big ball of pus. Those are A+ or severe abscesses. So as I said, this is diagnosed in the plant. In the processing plants, there are CFIA inspectors who are specifically looking at livers and grading them. Here we have an example of a large abscess on the surface of the liver. Here, the liver's been cut open and you can see there's an abscess on the inside of the liver. And this is an example of a liver that had an abscess that has since resolved, and it's visualized as a scar on the surface. So what is the actual cost of liver abscesses to the industry? The last time this was estimated was in the 2016-2017 Canadian beef quality audit. At that time, estimates based specifically on losses as a result of livers being downgraded or condemned was approximately $61 million per annum. Another important thing that this audit showed was that compared to 1999, the number of abscesses that we were seeing and the number of severe abscesses has increased quite substantially. But it should be noted that these numbers are based on liver costs and liver discounts. So these do not account for costs that are associated with reduced growth efficiency, increased feed costs, trim, increased trim of the carcasses. And, it's been 10 years since this was done. So it's important, I think, that we figure out how to do this estimate again to see, is this trend continuing, where we're getting a continued increase in abscess rates? And can we figure out what some of these unseen costs are? So a recent paper came out, I've got the reference here, Taylor et al. 2025, where they were looking at modeling costs in the US of liver abscesses. And they came up with a range of numbers, but I think the most confident number they came up with was approximately 256 million per year. But the important thing was that most of the losses that were associated with that number were before slaughter. So these are your increased energy maintenance. So a pen that has 20% of animals with liver abscesses will have 4% increased maintenance energy. Cattle with A+ livers have been a lower carcass weight, so approximately 29 pounds lower. And this varies by individual, but anywhere from $11 to $275 per animal. This figure shows the effect of liver abscess and the severity on carcass weight, dressing percentage, and carcass trim. So obviously the thing that's most clear here is that there is an effect, and that effect is most severe when we have A+ livers. And here you can see the increase in trim that is occurring on those carcasses when they're being butchered in the plants, to account for sometimes these abscesses will adhere to the walls of the carcass, and they need to cut away some of that to get rid of some questionable meat. So what factors influence liver abscess rates? One thing is that it's highly variable. So commonly the numbers that are thrown around as to industry standard are anywhere from 10 to 30% of animals will have an abscess. Number that we talk about in Canada is closer to that 30%. But this is going to depend on location. Some feed yards have significantly lower. Some feed yards have significantly higher rates. We've done experiments where almost all of our animals are abscessed. Steers are going to have more abscesses than heifers. This is related to intake. Dairy animals, so we're seeing a lot more beef on dairy. Dairy animals being incorporated into feedlots, they spend a lot more time on feed. So the more days on feed, the higher your rates of liver abscess. And then diets and composition of your diet can have an effect. The more digestible your diet is, the more likely the animals are to have gastrointestinal issues. So how do they develop? The currently accepted theory of how liver abscesses develop is that it's related to ruminal acidosis. And so the consumption of highly fermentable diets results in the rapid production of volatile fatty acids in the rumen. The rumen is only able to absorb those acids at a certain rate. So if you're producing more acid than the animal is able to use for growth, then the pH of the rumen is going to decrease. If it decreases to enough of an extent for a long enough period of time, you can get damage to the rumen layer, which can allow for bacteria in the gut to get into the rumen layer and into the bloodstream. And so this is called rumenitis. Once bacteria get into the bloodstream, they go through the liver, through the portal vein, where they have a potential to colonize and form an infection, which is effectively what an abscess is. But one thing that we know is that not all bacteria that get into the liver cause infections. We've detected the main organisms that are known to be involved in abscess formation. We found them in healthy liver tissue, and we found them in healthy livers. So one of the questions that we need to figure out is what's happening to those animals that are developing abscesses? Is there increased stress? Is there other factors that are involved in the ability for the organisms to establish infection? But so that was kind of the classical theory, a widely accepted theory of how abscesses are forming, is it all involves the rumen. Now there's starting to be some thought that maybe there's other parts of the gastrointestinal tract that are involved. And one thing that you tend to see is that when there's acidosis in the rumen, you also get depressed pH in the lower gut, so hindgut acidosis. And what's my next slide here? So if we look at what the gastrointestinal tract of ruminant looks like, see we've got the rumen here, abomasum, small intestines, cecum, and colon here. If we look at the structure of the epithelial layer, you can see that the rumen is highly stratified. So there's multiple types of cells there, and it's a very thick layer. So this provides quite a lot of protection to prevent damage. When we're looking at the hindgut epithelial layer, it's a single cell layer with a layer of mucus on top. And there's also these specialized immune cells called goblet cells that might be a way that organisms can get across. So we do know that damage does occur in the rumen. We do know that that is one way that they can get in. What's less known is, does this hindgut acidosis also play a role? And if so, how much of a factor is it? It's a lot easier to do studies looking at rumen factors. You can cannulate the rumen, you can put things in the rumen. It's a lot harder to cannulate into the cecum or into the colon. So there just hasn't been as much work that's been done looking at that. Another thing that I think more recently has become appreciated is that liver abscesses are polymicrobial. So there's been a lot of work that's been done on abscesses and understanding liver abscess microbiology over the last 30 plus years or so. And the majority of that work has been done on the bacteria, Fusobacteria necrophorum. Now, this organism undoubtedly is one of the key players. You find it in every single abscess. But you also find a lot of other bacteria. So there's an organism called Truparella, which is commonly found in abscesses, Bacteroides, which I'll talk about in a few slides, Salmonella, Escherichia coli, which you guys have heard about. So you can also isolate these. It also makes sense that they might play a role, but the exact role that they play is not really understood. But I think what has become more apparent is that while you always find fusobacteria present, the one other organism that you tend to commonly see amongst abscesses is Bacteroides. The other ones are either regional, so Salmonella. There's a group in Texas that is really focused on Salmonella. They tend to find a lot of Salmonella in their abscesses. I've never seen it in any abscess I've worked with. There's a lot of other people in the field who have never been able to isolate it. So it may play a role, it may not play a role. So this polymicrobial nature of these abscesses brings about this idea of microbiomes. And microbiomes are bacterial communities. So in nature, most bacteria exist as a mixed community. And the definition of a microbiome is a microbial community consisting of bacteria, fungi, archaea, protozoa, viruses that occupies a defined habitat and has a distinct physiochemical property. So we've done some work. This is a figure that was taken from this review here by Pinnell and Morley, but we've found similar results, where if you look at the metagenomic profile of a liver abscess microbiome, each one of these colors in these bar graphs represents a different bacteria. And the size of that color is proportional to the amount of the bacteria. So the bigger, the more one size, the more purple there is, the more that purple bacteria, which is fusobacteria, is found in that community. So if you look at this, most of these bars are primarily purple, right? So fusobacteria makes up a large portion of the community in a majority of the abscesses. But there's a lot that also have this orange bar, and that orange bar is Bacteroides. So Bacteroides is quite difficult to grow. It's more sensitive to oxygen than fusobacteria is. And so I'm not sure if previous studies that have been working on this, if their inability to isolate this organism has been related to how they handled the abscess before they tried to grow things from it. So this is the approach that we've used in our lab. So this is a picture of a liver abscess that we collected and dissected. You can see it's a nice juicy one. We took the pus from that abscess and we either directly inoculated it into tubes and allowed it to grow, so we're enriching for whatever's in there that wants to grow, or we inoculated a plate and then made a dilution series, which is you're just serially diluting the amount of bacteria and then plating them. So we did all these experiments in a anaerobic chamber. So this is a picture of ours in the lab. These are the ones where you've got to put your hands in, and it's hooked up to gas tanks that have anaerobic gas in them. We incubated everything in this chamber, and then when we looked at our plates and found the ones that had things growing on them, just randomly pick colonies off that plate. So we knew that a lot of these were going to be fusobacteria. A lot of the colonies that we picked were going to be fusobacteria because that's the majority of what's there. So we used a quick screen to quickly identify what those fusobacteria colonies are, put them into one pool, and then anything that wasn't fusobacteria, I thought, well, at the time when we started this, they had just kind of discovered the fact that a lot of samples had high amounts of Bacteroides, but nobody knew what it actually was. So I thought, well, what if we look at all these things that are not fusobacteria and see if any of them are Bacteroides? So this graph shows the isolation of Fusobacterium from liver and rumen. And one of the things that I want to point out here is that in cattle with healthy livers, we isolated a fairly significant number of Fusobacteria from those samples. Livers that had abscesses obviously had higher rates, so that makes sense. But yeah, the main point I want to make here is that you find these things in healthy livers too. So, but what about these Bacteroides? So we were able to successfully isolate Bacteroides from liver abscesses. This is a scanning electron micrograph of our isolate. It's not particularly interesting looking. Most bacteria either look like circles or rods. But the first thing I wanted to know is, what species is this? So within Bacteroides, there's over 60 different species of Bacteroides that are known. And they have very different properties. Bacteroides is one of the most common organisms that you find in the gut. They're a central player in carbohydrate digestion. But if you can figure out what they do, or what they are, and sequence their genome, you can get an idea of what they might be doing. So I was pleasantly surprised to find that when we sequenced the genome of this isolate, it was actually a new species. So it hadn't been seen before, hadn't been grown before, hadn't been described before. And I've putatively named it with the help of ChatGPT, providing me some suggestions of good names for bacteria. Bacteroides purulensis. And purulensis is related to the fact that we got it from pus. So we're in the process now of publishing a paper to actually get this officially recognized by the microbiology bodies who decide whether or not it's real or not. But it's a heck of a process, I'll tell you. So why am I so excited about Bacteroides? So Bacteroides and Fusobacteria, this is like my pet hypothesis that I've been talking about for, since we did this work. And, I don't know, I might be full of it. At some point, somebody might disprove me, but for now, I think it's a good idea. So we did an experiment where we wanted to see if we grow the main organism, Fusobacterium, in the absence and in the presence of this other Bacteroides that we isolated, what happens? How do they grow? And when you do this, the main way or the easiest way to measure bacterial growth is just to measure how, it's called turbid, or the bacterial growth solution is. So here we have a bacterial media that is clear, see-through. It hasn't been inoculated with bacteria. And here we have one that has been inoculated with bacteria. And you can see that this is a lot more like cloudy. So that cloudiness, when you shine light through it in a spectrophotometer, the bacterial cells absorb the light. And so you get an increase in absorbance as there's more cells in the media. And that generates these characteristic curves where you get this rapid increase as the organism is growing exponentially. And then it levels off and it starts going down as here your rate of cell growth and death is equal, and here your rate of cell death is greater than cell growth. So what does this graph show? So in the blue is our growth of Fusobacterium necrophorum. Down here is Fusobacter necrophorum growing alone, and the light blue is when it's growing in co-culture with Bacteroides. Pink is Bacteroides, again, alone is dark pink, and in co-culture is light pink. So when I saw this, I was very excited, and you might be thinking, well, what the heck does that mean? And what this said was that when you have these organisms together, they both grow better, particularly Fusobacteria necrophorum. So it grows to a higher level, so there's, and it grows a little bit faster that we've seen in some other examples. And I don't think I put the data on here, but the reason that this was really intriguing to me was when you look at the genome of this organism, it encodes a lot of things that would help break down tissue. So my hypothesis is that if you have both of these together, they both work together to help break down the tissue, colonize the tissue, and then over time, Fusobacteria grows much faster than Bacteroides. And so you get this out-competition of the Bacteroides to the Fusobacteria. So those abscesses that might be all purple or all Fusobacteria versus the ones that have both of them might actually be just differences in age, where you have this natural evolution of the community towards a Fusobacteria dominant community versus a mixed community. So there's my hypothesis. Reynold, we can talk later. So one of the other things that we've done in this project is we've looked at, okay, what happens to the liver function itself. So as I mentioned, what time am I? Okay. In most cases, liver abscesses are asymptomatic in the animal. So when we looked at, we did this both by taking liver biopsies, which Steve Hendrick helped with. I think I saw him here. And we also did this post-mortem where we collected the abscesses from the plant and just dissected them. And we looked at gene expression in the tissue associated with the abscess versus healthy tissue. And what we saw was that there was a significant change or a significant effect on gene expression in the liver tissue, and that a lot of the genes involved in metabolism were downregulated. And so the main role of the liver is to detoxify and metabolize things that come to it. So if you downregulate that, then that's an impairment of liver function. We also saw increased immune response and increased pathways associated with cell death. And so that presumably is the liquefaction of the cells as they're turning into pus. And also changes in the expression of genes related to something called the extracellular matrix. So a lot of the genes that are produced by this Bacteroides target carbohydrates that are on the surface of cells that help keep stitch cells together into tissues. So I think what they're doing is breaking down those kind of the cellular glue that holds the tissues together, and helping digest the tissue, which I think would help initiate Fusobacteria infection. All right. I have no idea how I'm doing for time here, but I know I'm behind. 20 minutes, okay. I never leave home without my coffee cup. It's kind of like my security blanket. Okay. So how do we prevent liver abscesses? Tylan. Tylan is the primary feed additive antibiotic. It's a macrolide antibiotic. It has broad activity against gram-positive and gram-negative bacteria. broad activity against gram-positive organisms, but limited activity against gram-negative. This is also something that's quite interesting, because the two organisms I was talking about are what are called gram-negative. It's related to the structure of the cellular membrane of the bacteria. So if the macrolide has limited activity against gram-negatives, why does it actually help to reduce liver abscesses? And I don't know, like we know that tylosin is one of the few things where there's really good evidence that it actually does reduce incidence rates of liver abscesses, but I don't think the actual mechanism of how that's happening is well understood. So there's this increased pressure to reduce antimicrobial use in livestock. And I think that's really where this work, I hope, will lead, is can we develop some approaches that will not be antibiotic use and can help to reduce that somehow. It's not an issue right now, but if at some point the regulators decide to make it an issue, then the incidence of abscesses is predicted to go up pretty substantially. And then that cost associated with liver abscesses is also going to go up. So ideally, we'd like to have some other options before that happens, if that happens. So these are just some of the numbers of how effective tylosin actually is. Doesn't affect gain or intake. It does reduce liver abscess by approximately 30%. Larger meta-analysis, again by this Taylor et al., found that if tylosin is taken out of the diet, then liver abscesses prevalence could go from what they found in that study of approximately 21% on average to almost double. Another common antibiotic that is seeing some use, it's not quite as common, is virginiamycin. Less is known about this. We have a project that is being led by Rahat Zahir and Tim McAllister at AFC, where we're going to do some trials with virginiamycin and just compare it to tylosin and see how it is. Let's skip over that. So, what are some other ways that we can control? Feed management and adaptation to diet is one of the important things to reducing the risk of acidosis. So given the hypothesis that ruminal acidosis, potentially hindgut acidosis, increases your susceptibility to liver abscess, then doing things that will reduce the risk of acidosis can hopefully reduce liver abscess. There are a lot of non-antibiotic approaches that have been tried. These are not exhaustive in nature, but people have tried looking at increasing the amount of fiber or forage in diets. Theory is that, well, you've got less concentrate, but you also get more rumination, so saliva will help buffer the rumen pH. There was a fusobacteria-specific vaccine that was developed years ago. Didn't seem to be all that effective, and I don't know if it's still on the market. There's a number of yeast-based products that are being sold that are supposed to help reduce abscesses. Direct-fed microbials, essential oils. when you look at the data, the results as to whether or not many of these things actually work is inconsistent or inconclusive. So the one thing that is pretty well established is that continuous inclusion of tylosin into rations is the most effective way. So this brings up, why are the results so variable? Doing these types of studies is very difficult. The effects that you see from a lot of these treatments is quite small. And the smaller your effect, the more animals you need in an experiment to actually reliably measure that with statistical significance. So you need a lot of animals when you're doing liver abscess trials in order to get statistically valid, consistent results. And most experimental feedlots can't house enough animals. Even if they do, often our budgets don't accommodate studies with thousands of animals. So for example, in Lethbridge, we have an experimental feedlot. Max number of animals that I could fit in there in a trial is 500. Now, how many animals are we talking about? So if we're looking at a baseline amount of liver abscesses from, say, 25% to 20, so a 5% reduction, we would need 1,000 to 1,500 animals per treatment. So you'd have your experimental treatment plus your control, so you'd need 2,000 to 3,000 head. If you've got a really big signal, so here we're looking at a 10% reduction, you can get that down to 300 or 500. So I think that's why a lot of these studies are consistently showing Thailand effects and Virginiamycin, because they're very large effects. But for things like the yeast extracts and direct-fed microbials and all these other products that are being sold, they may have an effect, but maybe that effect is smaller. So depending on your experimental design, some studies will find significant improvements, other studies may not. And I think my main point here is that we need to do it better. So how do we do this better? We need to get more samples, we need to get more animals. So it's fairly easy to collaborate with Cargill or JBS to get lots of livers and lots of abscesses. What's more difficult is how can we trace back those livers to metadata like animal performance, animal treatment, diets, etc. So there you need relationships with both the plants and relationships with producers. So the ideal situation is you can collaborate with a producer who might be able to run some sort of trial at a commercial scale. I have no idea how much this would cost, probably more than I have. And then you can collaborate with the plants to get access to those livers. Now you have really good data on the performance and the treatment of the animals. You have enough animals, and you have good samples at the end by getting the livers. So, final remarks. We can find liver abscess pathogens from healthy tissue and healthy livers. So there's something happening with the animals that develop abscesses that we don't yet understand, which is causing them to go from healthy to unhealthy. Dietary adaptation and diet formulation, feed processing are all important to reduce risk of acidosis. High stress, high competition may contribute to reducing the immune function of these animals. Feed intake fluctuations can lead to acidosis, which would be a problem. Currently, the only thing that I have seen data which is pretty convincing as a way to reduce as a way to reduce liver abscess is antibiotic, Tylan. But yeah, we need to continue trying to understand the mechanisms involved, both the microbial as well as on the host side. And we need to find ways to better design our trials so we can get enough animals to get some statistically significant results. So I just want to acknowledge your funding. So this work was primarily funded by BCRC, and Genome Alberta also threw in some matching money for that BCRC grant. AFC pays my salary, and we work with a lot of really great people, and I just want to specifically highlight Nora Chomistek, who's my main lab technician and works very, very hard and keeps me sane. Marissa McCormick and Leah Russo were also two technicians that were key to this work. So, and again, here's my email. Happy to, any questions you have, feel free to email me anytime, and I will do my best to answer as best I can.
Should have time for some questions. Questions from the audience?
I'd actually like to start. I have the microphone. Rob, you talked at the very beginning of your presentation about a potential like live animal test, right? I could see the value in that from the research perspective, for sure. But I'd like to throw it out to the audience if any of the producers in the room see a value in that from their end.
Do you have any data or information on how that is affecting the liver abscesses? Are we seeing an increased level in these heavy cattle, or are things kind of, the percentage remain the same?
I have not seen, I would say the closest thing I've seen to data related to that is the beef on dairy animals, because those animals are on feed for, say, an extra 100 and some odd days relative to an Angus cow. And you see significantly higher rates in abscesses in Holstein cross breeds. Now, how hard you push, so if we look at things like, we've done experiments looking at using wheat as the primary grain as opposed to corn or barley, and my understanding. So I will preface this and say that I am a microbiologist, not an animal nutritionist. So, but wheat, I believe, is much more fermentable, and you have a higher risk of ruminal acidosis feeding high wheat compared to some of the other grains. And in those, again, most of the experiments that we've done have been quite small, and we've seen really variable amounts. So I'm not sure if I have the best answer for you.
Okay. Anything online?
Yeah, corn would be quite a bit slower, at least if it's ground. If it's flaked, it really depends on the flaking density. And if people are using high moisture corn or snaplage, fermentation time impacts rate of degradation. But we would almost always put wheat faster than any processed corn.
Foucan said, is there any research focusing on developing new ATB alternatives to replace tylosin? For example, what if someday the government bans tylosin?
Yes, that's kind of the goal of the research is, can we find some other ways? Potentially, I think the most feasible would be if we can figure out how to make a vaccine that actually works well enough that it is cost effective and makes sense to use. So I have some ideas on how to do that, and maybe targeting more than just fusobacteria. But yeah, again, I'm not really an expert in vaccine design, so it's still at the idea stage right now.
Just a question out of my ignorance, I guess. I assume by this gentleman's comments that you don't get what you get paid on fat cattle doesn't change if their livers are condemned. Is that correct? You're paid the same money if the liver's condemned or not condemned, correct? So, you talk about economics. How you're going to sell to me as a feedlot operator is what are the subclinical advantages of controlling the abscesses? Because if what I get at the packing house doesn't differ, why would I be interested other than if there's a change in average daily gain or, time to finish?
Just to add on to that, if you're trimming out, if there's abscesses on a carcass and they're trimming the animal more, obviously you're going to reduce the value that you get out of that carcass. That would be a huge thing. But even for us to understand the impact on feed to gain and average daily gain would be key. And like I am actually, when you put the slide up there that we haven't done a beef audit for 10 years, kind of makes me think, why are we not doing it? Because I would guess that the impact of liver abscesses have gone up by 5 times in value with the extra value of the carcass, and I could only assume a much higher rate of liver abscesses.
Yeah, I think that's the key thing is although there's some numbers that have been estimated in the US as to what that unseen cost is on performance, we don't know that number in Canada.
Dr. Joyce says, would you recommend, and I apologize, I'm just a poor photographer, would you recommend chlorotetracycline instead of liver abscess control instead of tylosin, since not has an important antimicrobial based on HCVDD classification?
Okay, I can clarify that one. Yeah, there's also a lot of random. So the question is about using chlorotetracycline instead of tylosin because it, in theory, has a lower impact to human health based on the Veterinary Drug Directorate classifications currently.
Yeah, again, I'm not qualified to provide advice on what antibiotics to use in your ration, so.
And I think just a point around the beef quality audit, I think there may be some plans afoot to redo that coming up, potentially.